Rahim. This is Dr. Nighat Aslam from Department of Biochemistry. This is my third lecture regarding molecular biology techniques. In this lecture, we will study about what are the probes and what are their applications in medical sciences. You have studied about the use of restriction enzymes. Restriction enzymes produces a large number of DNA molecules by cleaving the DNA. So, how the DNA sequence of interest can be picked out from such enormous amount and such enormous array of fragments? The answer lies in the use of probe. And what are probes? Probes are short piece of single-stranded DNA or RNA labeled with radioisotopes such as radioactive phosphorus, or with non-radioactive molecules such as biotin or some fluorescent dye. So these short piece of DNA or RNA, which can be either labeled with the radioisotopic or non-radioisotopic molecules, they are called the probes. The sequence of probe is complementary to the sequence of the DNA of interest, and so these probes, they are complementary to the to the sequence of interest, so we can pick out from our DNA or fragment of interest from large array of fragments which were obtained from the restriction enzymes. So these probes, they are used to identify which band on a gel or which clone in a library contain target DNA. So this is the probes, they are used in the identification procedure, and this identification procedure is called the screening. This is the how we can use to identify our DNA of interest. The first of all, this is the agar plate, Petri plate, which contains the agar as the growth media, and on this growth media bacterial colonies, these blue colored bacterial colonies, they are grown. We transfer these colonies on this is the nitrocellulose paper. We transfer these blue colored colonies on this paper. This is not a Petri plate, this is a nitrocellulose paper. We press this nitrocellulose paper onto the plate, and cells from each of the colony adhere to this paper, and this is the orientation marker, so that after the end of the experiment we can identify that these bacterial colonies were originally starting from these colonies. So this is the orientation marker. After transferring the colonies on the nitrocellulose paper, we treat this paper with alkali so that the DNA can be denatured, and heating is applied so as to fix the DNA in place. After fixing and denaturation, the radioactive DNA probes, they are applied to this paper, and incubating it and then washing to remove the probe that has not hybridized. The probe will not hybridize to every colony present on the present on the plate. It will be only hybridized to the specific DNA sequence. So the other probes, they are washed away. As these probes, they are radioactive, so we do the autoradiobiography, and it is exposed to the X-ray film. So we can see that here on the X-ray film only our probes, it has been probed to the two colonies only, and this is the orientation point. So by going to the start, we can find out that our DNA of interest is present. Our DNA of interest is present in this colony and in this colony. So this is how the probes, they are used to identify the sequence of interest, the DNA of interest. And this is called the screening procedure. From the sequence of all or part of the DNA, if we know the sequence of all or part of the DNA, we can synthesize the oligonucleotide probe. If we don't know the sequence of the DNA, we can use the amino acid sequence, and this amino acid sequence can be employed by using the codon for that species. So oligonucleotide probe can also be synthesized by using the amino acid sequence. But in this case, as you know that there are different codons for more than one codon for one amino acid, so a set of or a mixture of oligonucleotide probes will be synthesized. If the sequence match exactly the probes, which is 15 to 20 nucleotide long, it will be hybridized. What are the cDNA probes? cDNA probes, they contain thousands of the bases. Their binding is not affected by single base gene. So if the bases they are changed or there is mutation, the cDNA probes will be bound with the original DNA and also with the mutated DNA. So it cannot differentiate between the single base changes between the DNAs. cDNA probes can be used to detect DNA fragments on the southern blot transfer and to detect and quantitate the RNA on the northern blot transfers. Oligonucleotide probes can detect the mutations in the sickle cell mutations in the beta globulin genes. DNA isolated from our white blood cells. First of all, DNA is isolated and then it is amplified, then is denatured and applied to filter. Now on this filter we will apply our oligonucleotide probe. A new oligonucleotide probe is synthesized. Oligonucleotide probe is synthesized to a point mutation, so it will be only bound to that DNA which will be mutated. At this mutation, you know that the glutamate, it is changed to the valine in the sickle cell gene, and at the position 6 in the patients with the beta S gene, it is applied to the filter. So the probes, they will bind to the mutated genes. DNA isolated from the heterozygous individuals, which are sickle cell trait, or homozygous patient sickle cell disease, contain a sequence that is complementary to the probe. So to this probe, which is synthesized complementary to the point mutation, it will bind to both of the sequences. That is, it will bind to the patient's DNA and it will also bind to the heterozygous individual. A double-stranded hybrid forms that can be detected by electrophoresis. This is the how the allele specific oligonucleotide probes, they detect the hemoglobin S allele. First of all, the portion of the gene of the beta S, this is the portion of the gene, this is the portion of the gene for the beta S chain of the hemoglobin S. DNA codes for valine instead of glutamate in the 6th position of the beta globulin. This is the probe which is specific This is the upper DNA part. This is the probe, and it is specific, and it will bind. It is complementary to the DNA fragment coding for the hemoglobin S. So it will bind to the mutated DNA, and the normal DNA from the normal detail. As our probe, it contains the T. It will not bind. It is not completely complementary to the DNA fragment coding for the HBA. So if our probe, it will bind and it give the fluorescent, we can detect from the X-ray film. And if our nucleotide probe, it failed to hybridize with the DNA fragment, we will not have any fluorescent, we will not have any fragment, any stain on the X-ray film. If we use a pair of allele specific probes, a pair, one specific for normal allele and one specific for mutant allele, it will allows all three possible genotype, which are the normal, homozygous normal, heterozygous, and homozygous mutant. It can be distinguished. ASO probes can only, allele specific probes are useful only if the mutation and its locations are known. See, this is the there are three spots of the DNA. One is the homozygous normal, the second one is heterozygous, which is carrier, and the third one, this is the third one, this is mutant. So we use the probe, mixture of probes. This row, the first row, this one, this row probed with ASO specific for a normal beta A gene. So the probes which have the sequence complementary to the normal gene, it will bind both, it will bind both to the homozygous normal and heterozygous. And the third, which is the mutant, which is affected, which is the DNA from the patient, it will be not bound. The second row, this one, this row was probed with the ASO specific, which was designed against the mutated genes. It will bind to the heterozygous and it will bind to the homozygous mutant. The normal DNA, this is the normal DNA, it will be not bound with the probe. So in this case, we can determine that we can find out the heterozygous mutant allele, heterozygous carrier allele. What are biotinylated probes? These probes, they were designed so as to get rid of the use of radioactively labeled probes, as the disposal of radioactively labeled probes, their waste, they are becoming very expensive, and also if they are not wasted properly, their radioactivity, it can damage it can be hazardous to the human health. So the non-radioactively labeled probes, they were developed. How they were used? A DNA fragment, first it is displayed, for example, on gel electrophoresis, it hybridized with biotinylated probes. These are the probes which were attached with the biotin. Biotin was chosen because it will bind with the avidin, and this avidin, it is attached to the fluorescent dye, and which can be this can be visualized. So they can be made visible by immersing the gel in a solution of dye coupled with avidin. So the probes which were attached with the biotin, they can bind with the avidin which is attached with the dye. So after washing away the excess evidence, the DNA fragment can bind the probe if it is fluorescent. What is in situ hybridization? In situ means we will find out, we will detect the DNA in the cell and in the tissue preparations directly. We don't know, we do not isolate the DNA. We detect the DNA, we localize the DNA within the cell and within the tissue preparation. This is the in situ hybridization, a process in which fluorescent probes can allow detection and localization of DNA or RNA sequence in cell or tissue preparations. If the probe is fluorescent, this is called the FISH, means fluorescent in situ hybridization. Antibodies. If we don't know the sequence of the DNA, and also we don't have any information about the sequence of amino acid, what we will do, that we will get the we will clone the cDNA in expression vector and allow the cloned cDNA to be transcribed and translated. Now we have the proteins. From this protein we will get the antibodies. These antibodies can be labeled and they are used to identify which bacterial colony produces the protein and therefore contain the cDNA of interest. So we can find out the bacterial colonies by attachment, by treating them with the labeled antibodies. So this blotting and hybridization techniques, blotting and hybridization techniques allow the visualization of specific fragments. Southern blotting. First of all, we will study about the southern blotting. This is a technique that combines the use of restriction enzymes, electrophoresis, DNA probes, and by using all these techniques, we separate and detect a piece of DNA. Southern is not originated from the word south, but it is obtained from originated from the word from its inventor, who is called Edward Southern. This is the name of its inventor who invented this blotting technique. We will study about this blotting technique by using this figure. First of all, these are the two persons. We obtain the DNA from their white blood cells. So we have the two different types of the DNA, and when we cleave these DNA, with the same restriction enzymes, their pattern of restriction is different. The DNA fragments that will be recognized by probe hybridize DNA from person 1 show a restriction not present in the person 2. You are seeing that the person 1 has extra, this person 1, it has the extra restriction site. So we obtain two types of the fragments, and here we have only one type of fragments because this person, it lacks the restriction site which is present in the person 1. We do the electrophoresis. Of these DNA which were restricted, and denatured the DNA into the single stranded and transferred by blotting action to the nitrocellulose membrane so as to for the further analysis, creating a replica of the gel. So this blue color, it is the gel, and it is transferred to the nitrocellulose membrane. Now this is hybridized with P32 labeled probe. Now you are seeing that the person 1, it has the different pattern of the DNA probing, and the person 2, it has the different probing with the on this hybridization technique. So we can differentiate between the DNA of the two persons, because these two persons, they have the different restriction sites, and this differentiation can be due to the usage of combined uses of restriction enzymes, DNA probes, electrophoresis. And so this process by which we use the DNA, we do the restriction restriction digestion, and then we do the electrophoresis. Then this electrophoretic material, it is transferred to the it is transferred to the nitrocellulose membrane, and this nitrocellulose membrane, it is then radioactive probes. By using the radioactive probe, it hybridizes to the restriction fragments A and B and C, producing dark bands on exposed X-ray film. So the DNA fragments of two individuals differ in the region recognized by probe. This alteration in the DNA could reflect a disease-producing mutation. So a disease mutation, they will also have the different pattern of its probing, or polymorphism that may or may not be linked to clinically important alteration in the DNA. This difference in the DNA of the two persons can be applied in the forensic sciences. For example, we have the electrophoresis of the two suspects. Electrophoresis of the DNA, this is the suspect 1 and this is the suspect 2, this is the victim, and these are the DNA markers in order to find out the size of the DNA of the fragments. So as we have studied before, that the DNA of the two individuals, they can be different, their restriction pattern, they can be different. By comparing their DNA to the evidence, by comparing the DNA of suspect 1 and suspect 2 to the evidence, we can find out that which one is the guilty. Here we can, after doing all the procedures in the southern blotting and exposing the X-ray film to the membrane, we find out that this suspect 1, it matches with the evidence, and this suspect 2 is free. Now we can find out that this suspect 1, this is the original criminal, because its DNA sequence, the after getting from the treating with the restriction enzymes, their DNA pattern is similar to the evidence. So this suspect 1 is the original criminal. So this technique, it can be also employed in the forensic analysis. So this is the application of the southern blotting. There are other blotting techniques also. And this is the southern blot, which we have studied by using the DNA, then do the electrophoresis and probing. Now what is the northern blotting? This is similar to the southern blotting, but here we use the starting sample RNA instead of DNA. So if we use the RNA as starting sample, and then other procedures, they are the same, like electrophoresis, probing and exposing to the X-ray film. This is called the northern blotting. And what are the western blotting? In western blotting, we use the protein as our sample. Again, they are electrophoresis, electrophoresed, and in the western blotting, we use antibodies as the probes. There is another fourth hybridization technique, which is called the southwestern blotting. It examines the interaction of the protein and DNA. In this method, proteins are separated in the same way by the electrophoresis, blotted to a membrane, denatured and analyzed for an interaction with particular sequence by incubation with specific labeled DNA probe. This is the southwestern blotting. So this is all about my today's lecture. Best wishes. Allah Hafiz.